think we can start with the first speaker of today, which is Alex Francette. Alex studied at the University of Pennsylvania, where he graduated in molecular biology and biochemistry. And since 2018, he's a PhD candidate in the lab of Karen Aren at the University of Pittsburgh, where he's now working on the role of Paf1 on transcription. And I guess we'll hear more about it in a moment. Alex, we're really happy to have you here. The floor is yours.
All right. Well, thank you for that introduction. Today, I'll be telling you a bit about a critical and conserved elongation factor known as the Paf1 complex. So today, I'll give you a bit of background, talk about experimental setup, and here you see Paf complex subunits popping on and off of RNA polymerase. And I'll tell you a bit about my observation of Paf1 complex's roles in transcription regulation, elongation, RNA splicing, antisense transcription, before diving a little bit into the role of a Paf1 complex dependent post-translational modification, histone K36 methylation, in mediating some of the phenotypes that we observe in the absence of Paf1. So Pol2 has its work cut out for it, as it needs to navigate the chromatin template, which is studded with nucleosomes, associate with factors that write, erase, and interpret epigenetic information, and coordinate with RNA processing enzymes to promote capping, splicing, cleavage, and polyadenylation of the nascent transcript. Thankfully, there are lots of proteins that are play a role along the way to help Pol2 do its job. And we're very interested in understanding what these factors do, through what mechanisms, and why. So now enter the star of the show, the Paf1 complex, which in Saccharomyces cerevisiae, the model system in which I perform all this work, it consists of 5 subunits, including Paf1, CTR9, RTF1, CTC73, and Leo1. And together, they've been implicated in a variety of co-transcriptional events, including transcription termination, export of mRNAs, recruitment of chromatin remodeling factors, transcription elongation, and very importantly, post-transcriptional modifications of both RNA polymerase II through serine 2 phosphorylation of the C-terminal domain, and histone modifications H2B monoubiquitylation, and H3K36 di and trimethylation. Now, we're very interested in understanding how the subunits of the Paf complex contribute to transcription and transcript regulation. And so answer how they contribute to transcription, we're taking a two-pronged approach. On one hand, we can look at classic deletion strains. You just knock out the gene of interest and ask how transcription changes. But on the other side, we can rapidly deplete proteins of interest using the oxo-inducible degron, or AID, system, which we see C-terminally append to our subunits of interest along with a V5 epitope tag, which allows us to add the drug auxin and induce rapid degradation of our proteins. And these both have their advantages and disadvantages. For deletion strains, we have cells which are in dynamic equilibrium. They've reached steady state. This allows any sort of long-term accumulative effects to accumulate and manifest. But on the other hand, any indirect consequences of Paf1 complex loss, for instance, this is a transcription elongation factor associated with Pol2, wherever you see Pol2, very easy to imagine how misregulation of transcript regulators can beget more misregulation. And so that muddies the water a little bit. And we don't, with our depletion system, we get to avoid some of that. We rapidly deplete, we see the immediate consequence of subunit loss before those more long-term indirect consequences start to accumulate. But unfortunately, any sort of cumulative effect of Paf1 that it's participating in isn't really observable with a short, rapid depletion. So we really believe that these two methods work very synergistically together and provide a much more holistic view for how Paf1 complex regulates transcription. So to validate the system a little bit, we can look at a Paf1 AID strain. So this is looking for the Paf1 subunit specifically, and track the V5 epitope, looking at Paf1 levels over the course of 60 minutes of auxin treatment, and see it's quite rapidly depleted. And treatment with vehicle does not deplete the protein of interest. H2B ubiquitylation is rapidly turned over with depletion of Paf, unlike K36 trimethylation, which seems to hang around for a bit. And if we extend this to the rest of the Paf complex, we see some pretty much expected patterns. They're all quite rapidly depleted. And so at the 30-minute time point, we have near maximal depletion for each subunit. And so that is the time point which I perform all of my subsequent experiments. So every rapid depletion time point is going to be at 30 minutes, either depleted or undepleted. H2B ubicodylation shows its most rapid turnover upon depletion of RTF1, which makes sense because our lab has shown the histomodification domain of this protein is both necessary and sufficient to promote Paf1 complex mediated H2B ubicodylation. And Paf1 and CTR9 are the core scaffolding subunits of the Paf complex, without which the complex cannot assemble. So H2B ubicodylation is also quite rapidly turned over upon their depletion. The other two play a lesser role in H2B ubicodylation, and under no condition do we see K36 methylation bulk levels change upon depletion. So now, using this system, we can assess the short-term consequences, either comparing depleted versus undepleted, or the long-term consequences comparing the null strain to wild type, and subject them all to nascent transcriptomics through 4'-thiouracil sequencing. And in this method, we are measuring 5 minutes of RNA synthesis, subject to a small amount of decay, but mostly dictated by synthesis rates. And we can ask, do these subunits differentially contribute to transcript expression? And so first, we'll take a peek at the null cells. So we're comparing null versus wild type, and I'm showing you heat maps of full change of 4TU output over coding transcripts from the transcription start site to the cleavage and polyadenylation site. So they're all sorted by gene length here, and blues mean downregulation, red means upregulation. And hopefully you can quite immediately see different subunit specific effects. I'm not going to get too deep into the nitty gritty here, but we see up and down regulation. We see subunits showing individual profiles. And now I want to contrast that with what we see with a rapid depletion, which is quite a lot of blue. Rapid depletion of Paf, CTR9, CDC73, or Leo1 seems to downregulate transcription, whereas depletion of RTF1 shows actually a mild upregulation. But overall, I hope this really impresses how different these profiles are from the immediate loss to the long-term downstream consequences. And so again, there's a lot of really rich information here, but I want to zoom in a little bit on CTR9 rapid depletion, where I think it's quite neatly exemplified. But if you go from the TSS to the cleavage and polyadenylation site, you'll notice that the blues get bluer, which is indicative of a loss of 4TU output as Pol2 is progressing through the gene body. And so as an elongation factor, we want to ask if Paf1 complex subunits affect transcription elongation dynamics. And so to look at that, we're borrowing a method from the Wolf lab known as the completion score, where you essentially take a sample of nascent transcription from the 5' and 3' ends, so A and B here, and we pretty much average the 4TU signal in either of these categories, these bins, and then we take the ratio of B to A for every gene. And this pretty much measures how much flux of 4TU that starts at the beginning of a gene makes it to the end of a gene. And we push this the sampling region in by 200 base pairs on either end to avoid any sort of edge effects. And so you'd imagine, just like a tightrope walker with an unsteady step, the longer the track, the less likely you are to make it from the beginning to the end. And indeed, if we separate our transcripts by gene length, and we ask what the fold change in completion score is between our mutant and wild type conditions, we see that the longest genes show the strongest loss in 3' read density by 4TU-seq in a Paf1 null condition. And this is consistent with a processivity defect, Pol2 not making it from the 5' end to the 3' end. Looking at the rest of the complex, we see that CTR9 and CDC73 also show evidence of a processivity or at least completion score defect, with the most extreme effects being Paf1 null and CTR9 null. So what about a rapid depletion? We can add auxin to a wild type strain and it doesn't affect completion score. That's really good to see. But rapidly depleting subunits of Paf1 complex show very similar profiles to the null. So the total transcriptomic profile I showed you earlier is vastly different, but this elongation dynamic defect is seen both in the rapid depletion and the steady state, suggesting this is a direct role of the Paf complex in regulating elongation. And so one process that we saw pretty immediately, very obviously being dysregulated was transcript processing. And so I want to talk about that pretty briefly. So if we just look at the Paf1 null condition for some of the few intron-containing genes in the yeast genome, we see that the relative levels of intron intron reads relative to the neighboring exons is much higher in a Paf1 null compared to a wild type. And we see that with CTR9 null, again, like very obviously by eye. And so I wanted to quantitate this and say, figure out, how pervasive this effect is. And to look at that, we can pretty much draw a line through the 5' splice site and ask what fraction of these reads remain unspliced in our various conditions. And what we see comparing our wild type to our Paf1 or CTR9 null is that just about every intron containing gene shows an increase of unspliced reads at the 5' splice site. And this is also seen at the 3' splice site, but I'm just showing you 5' for now. For the other Paf complex subunits, we see a more modest effect. And this is all in the long-term steady state conditions. So do we see the same thing upon rapid depletion? And the answer is no, not really at all. So undepleted versus depleted for any Paf1 complex subunit does not show a similar increase in intronic reads. So this is suggesting that Paf is not directly interacting with the spliceosome. It's not directly regulating transcript splicing, but this increase in unspliced transcripts occurs downstream of the initial perturbation of Paf1 loss. Okay, and so we're going to look at one more angle, and that is an antisense transcription. We're going to ask, do we see misregulation of antisense transcriptions in the absence of Paf1? And particularly, we're going to look at a class of transcripts defined by their upregulation in the absence of the methyltransferase that deposits H3K36 methylation. So set2 deposits H3K36 methylation, and so these set2 repressed antisense transcripts are upregulated in its absence. And in fact, they're upregulated in the absence of any Paf1 complex subunit in the null conditions. But again, this is one of those long-term effects that only shows up in the null and is not immediately apparent upon rapid depletion of Paf1. So we can ask, is there a post-translational modification? H3K36 in particular, is it contributing to some of these transcript regulation phenotypes that we see in a Paf1 null? So set2 in yeast deposits all levels of K36 methylation. And as I showed you, it is responsible for repression of antisense transcription. And this happens through recruitment of RPD3S and subsequent histone deacetylation. But it's also been implicated in recruitment and activation of the spliceosome by Tracy Johnson's lab and others. And we're not really sure how this interacts with elongation dynamics. But if you remember, our rapid depletion system retained K36 methylation at the 30-minute time point in which I perform all of these assays. And there's a key difference between the null strains in which K36 methylation can be completely lost, or at least trimethylation and a bit of dye. So we can, using yeast as our model system, very tractable, we can ask very specifically, does K36 methylation play a big role in mediating some of these Paf1 complex dependent phenotypes? And so to look at that, I generated an array of strains with varying levels of methylation or Paf1. For instance, we have a Paf1 null which pretty much loses all of its K36 trimethylation, at least by detectable by Western blot, and a bit of dye. And then we can look at the set2 delta 3 allele, a hypermorph discovered by the Winston lab, which is overactive, will deposit more methylation. And in fact, in the absence of Paf1, we can stimulate deposition of K36 trimethylation and restoration of a good amount of dye. And we can ask, does this suppress any Paf1 null phenotypes? And then on the other hand, we can look at loss of function mutations where we completely knock out set2 and all K36 methylation, or a hypomorph from Brian Strahl's lab, which shows a similar level of tri and dimethylation to a Paf1 null without the extra baggage of losing the Paf1 complex. And so we can ask, can any of these either suppress or phenocopy some of the Paf1 null phenotypes we observe? And first, I'm going to draw your attention to the completion score here on the right, and we can see losing methylation and the loss of function mutants here does not seem to affect completion score. And so it doesn't phenocopy Paf1 null, and adding methylation cannot suppress the completion score defect of a Paf1 null either. On the other hand, splicing shows a slightly different story, where loss of methylation does seem to increase relative fraction of unspliced reads, but it doesn't increase it to the level of Paf1 null, so it does not completely explain the degree of intron retention we see in Paf1 null. And indeed, adding more methylation back is not sufficient to suppress any of the unspliced transcripts here. So we can't really pin either of these phenotypes to K36 methylation, but we can pin it to antisense transcription. So if we compare, this is another Paf1 null strain, independently generated strain, new 4TU-seq data showing the same result, which is increased SRAT expression. And we see the loss of function set2 mutants show increased SRAT expression as expected, as we would hope, right? Given that they're defined by their upregulation in the absence of set2. But adding methylation in the absence of Paf1 for H2K36 seems to be sufficient to suppress expression of SRATs, almost all the way back down to wild type levels. So hopefully I've made a case that it's very informative and gains a lot of utility by comparing a rapid depletion short-term absence to the long-term absence. We get to see both sides of the equation. A rapid depletion of Paf1 complex subunits caused that immediate loss of transcript synthesis, which settled into a phenotype of both up and down regulation. Our elongation defect seemed to be a direct function of Paf1 complex activity, immediately impact upon loss of Paf1, not impacted by K36 methylation whatsoever. Accumulation of unspliced introns only shows up after long-term loss of Paf1 and is poorly explained by K36 methylation as well. Unlike antisense transcription, which is both phenocopied by the loss of set2 and suppressed by increased deposition by K36 methylation. With that, I'd like to thank the ARNT lab, this team that helped all of this work very smoothly, my committee and various other labs which have provided input, and the sources of funding that help keep the lights on. And I'd like to open up for questions.
Wonderful talk, Alex. Very clear and linear. Very nice. So we already have one question from Nathan Gamara. So, do you believe the chronic effect of Paf1 depletion on splicing is due to elongation or early termination effects? Or in other words, transcripts are not spliced because they have not transcribed the 3' splice sites?
Well, we see this on both the 5' and the 3' splice sites. No matter which angle we look, we see it. And upon rapid depletion, if it's a direct effect of Paf1 affecting elongation, we start our 4TU-seq labeling after Paf1's gone. So if there's early termination or elongation defects that are immediately directly related to Paf1 activity, we'd expect any sort of phenotypes for splicing to also be evident at that time. It's not out of the question that there are further elongation defects that happen longer term upon Paf1 absence. Again, not directly related to Paf1 activity. So yeah, I hope that answered your question.
Okay, thanks. So Damien Parrello has a question. You should be allowed to unmute now.
Hi, hello. Thank you for this talk. So I'm interested in Pol2 posing, and so I was just curious if you saw any changes in posing after the depletion of Paf1 or even after auxin, if you saw something.
Yes, great question. And in yeast, there is no evidence for Pol2 promoter proximal pausing. And I view that as a bit of a dual-edged sword. So on one hand, that's a difference between yeast and metazoans, but we get to look at the other effects of Paf1 outside of this very early effect that can further propagate across the rest of the gene body in the absence of Paf1. So we get to look at other Paf1 activities outside of pausing. Yeah, yeast does not have pausing.
Okay, thank you.
Okay, yeah, Ben has a question as well.
Hi, Alex, just beautiful work. Really, really, really great. I like the be able to look at the long-term loss as well as the acute depletion. My question is, what do you have any further thoughts on the RTF1 diagram and why that one's different than all the other subunits?
It's really interesting because if you look at null cells for Paf1 and RTF1, they're both a little bit sick to get either on their own. But if you put a RTF1 null in the context of a Paf1 null, it actually is a little bit healthier. Grows a little bit faster. And I think that argues that there are some Paf1 does a lot of things, and I think some of these activities work perhaps counter to each other and are a little bit modular depending on the Paf1 complex subunit we look at.
Awesome. Thank you.
All right. So thanks again, Alex, for the wonderful talk. And I'll hand it over to Ben for the next one.
Thank you.